In this study, we follow babies and young children with NF1 as they grow to better understand how areas like attention, learning, movement, and social development unfold across early childhood.
So I work in an animal model for neurofibromatosis, and I'm trying to understand what happens in the brain.
I'm a third-year PhD student at the University of Manchester, and it's such a pleasure to be here with you all today. And I'd really like to thank NTUK for inviting me to talk to you all. So I'm just going to share some of the work our team does with children and young people with NF1. Specifically, I will talk about 2 ongoing research projects, and this will be a really brief presentation. Okay, so the first project I want to discuss is called EDEN, which stands for Early Development in NF1. In this study, we follow babies and young children with NF1 as they grow to better understand how areas like attention, learning, movement, and social development unfold across early childhood. So one of the really exciting things about this work is that infancy and toddlerhood are periods of enormous brain development. Small differences we see early on can have cascading effects and can help us understand how children experience and interact with life later on. And importantly, it can help identify areas where support may be most beneficial. At this stage in life, many behaviors are only just beginning to emerge, which means that we can sometimes detect very subtle developmental patterns long before anything would necessarily be clinically detectable or noticeable in everyday life. Importantly, this research is not about judging children against a fixed idea of what development should look like. Rather, children naturally develop in many different ways and at different speeds. What we're really trying to understand is the wide range of developmental pathways within NF1, because that understanding may help us recognize where support could be most useful. And as for our second project, this focuses on young people receiving a treatment called MEK inhibitors, which I'm sure many of you are already familiar with. So this includes drugs like selumetinib, and we will plan on recruiting children on mirdametinib as well. As many of you will know, new treatments are increasingly being used to manage tumor growth in NF1. But families often describe effects that go far beyond physical symptoms alone. So treatment can influence energy, confidence, participation in school, and everyday quality of life. So alongside studying areas like movement, learning, and behavior, we also want to understand the lived experiences of children with NF1 directly from young people and families themselves. So our 2 projects connect together in that the EDEN study helps us identify very sensitive early developmental measures, such as patterns of cognition and behavior, and this gives us an opportunity to explore whether MEK inhibitor treatment may influence some of those same systems later on. Now I'd like to introduce the EDEN study in a little more detail. So how do we actually study the development of babies and toddlers? To investigate this, we have families visit us at the Baby Lab in London at 5, 10, 14, 24, and 36 months of age. Across these visits, we use a range of different tools to build what we call a developmental portrait. Really, a lot of what we do is just about paying very close attention to tiny behaviors that we all have but don't notice. So let me start with a quick question. When a baby can't tell us what they're thinking, what do you think we look at instead? Well, we become detectives of behavior. So where do babies look? What captures their attention? How quickly do they shift focus? A lot of our tasks are designed like very simple games. We might show them pictures, sounds, toys, and videos, and just see how they respond. We also use parent questionnaires, which are incredibly valuable because parents see development across far more contexts than we ever could in a single visit. Things like sleep, mood, sensory responses, and everyday behavior at home. We also have some more brain-based tools. One of these is EEG, which you guys might know already, which is just a safe and non-invasive way of measuring the brain's natural electrical activity. And I sometimes describe it like this. So if the brain were a football stadium, EEG is a bit like placing microphones outside the stadium. We can't hear individual conversations, but we can pick up the overall energy, rhythm, and activity of the crowd. Another important tool we use is called eye tracking, and this one is very visual. It tells us exactly where babies are looking on a screen, almost like a real-time map of attention. And just to make this more concrete, if I asked you right now to look at a busy scene, your eyes would naturally jump from one thing to another. Babies do exactly the same thing, and we're just measuring it very precisely. We also use a technique called accelerometry, which is like a small movement sensor, a bit of like a research version of a Fitbit. This helps us understand activity patterns across the day. So how much babies move, how variable that movement is, and how it changes over time. And finally, we also use ECG, which measures heart rate. Your heart rate naturally changes when you're excited, focused, or emotionally engaged. So it gives us another window into how babies are responding internally to the world around them. So really the crux of this is no single measure tells the full story. But when we combine them, we start to build a much richer picture of early development than we could ever get with one test alone. Right, so what have we found so far? One thing I really want to emphasize is that many aspects of development in NF1 look very similar to what we see in typically developing infants. For example, we see similarities in areas like language, overall physical activity levels, and social functioning. At the same time, though, we do see some differences in how information is processed or how development unfolds over time. For example, using EEG, we've observed small differences in patterns of early brain activity by 10 months of age. In some attention tasks, infants with NF1 have shown faster eye movement responses, called saccadic reaction times, which essentially means they shifted their gaze more quickly towards visual information. Now, faster or slower does not automatically mean better or worse. The brain can just process information in multiple ways. And part of our work is understanding what these patterns might mean developmentally. However, we've also discovered that difficulties with sleep appear early in infancy, which is a really important area to focus on for early intervention, support, that kind of thing. And we've also found some early differences in how play interactions unfold, with parents sometimes showing a slightly more directive approach during play. Findings like this help us better understand how children and parents influence each other during development, and this may eventually help us think about ways to support enjoyable, responsive interactions for families if they so want that. So overall, what we're finding is not a simple story of impairment, but a nuanced picture of developmental diversity, with many similarities alongside differences in our NF1 cohorts. Now moving on to the next study, this is a pilot study and not a randomized control trial. I really want to emphasize that. So we are not prescribing treatment. We're just simply studying the experiences of development in families that are already on this clinical pathway. So we see participants before treatment begins, and then again at 1, 3, and 6 months post-MECI prescription. During those visits, we look at areas like attention, movement, cognition, behavior. Basically the same, we use a very similar battery to what we use in Eden. And the reason is so that we can kind of use the early markers, which we see as being most different in our early development cohorts, and assess whether we see the most benefit with MECI in these same domains. One reason that this research is really critical is because MECI treatments were developed primarily to target tumor growth. But families and clinicians are understandably very interested in whether these treatments also influence aspects that actually shape everyday life and functioning. And at the moment, we do not yet have any evidence about how MECI treats cognition, or rather robust evidence of how MECI treatment affects cognition and behavior more broadly. And equally importantly, we want to understand the lived experiences from young people and families themselves, which numbers and scores can't fully capture. So we do a lot of qualitative interviews as well. Right, and we have some exciting research opportunities. So one is the qualitative interview study I just mentioned. And this study focuses on daily routines, well-being, symptoms, and how treatment fits into the everyday life of young people. And some of the most important insights come not from graphs, like I said, but listening to people's experiences. We're also launching a new study of Manchester looking at early development in children with NF1, as well as other rare conditions. And this project builds on everything we've learned from Eden, but will use even more advanced technology and more detailed developmental assessments during infancy and early childhood. So the goal is ultimately the same: to better understand early developmental pathways and help identify areas where support may be helpful, personalized, and timely. So yeah, I would just like to end now by saying a huge thank you to you guys, because research like this is only possible because families give their time and trust to these projects, or often while already managing a great deal in their everyday life. We're also incredibly grateful to the families who've taken part in our studies and to NAV Tumors UK for their continued support. And if anyone would like to ask us any questions, learn more, or get involved with research, please email us at nfresearch@manchester.ac.uk, or just come up and talk to me, if you want to know anything more. Thank you.
Hello everyone, thank you for inviting me to give this talk. So I work in an animal model for neurofibromatosis, and I'm trying to understand what happens in the brain. So NF1, as we all know by now, is a genetic condition that causes a deletion in chromosome 17. So what this actually does is it affects the neurofibromin gene, and this missing piece actually alters how the brain develops and how the body develops. So on a very, I'm going to take my presentation on a very cellular level, and I'm going to try and keep it simple. So when we say neurofibromin gene is being affected, what actually happens on a cellular level is that the gene codes for a protein called neurofibromin, and that is useful in a cellular signaling pathway. So the most common pathway is a RAS pathway, which actually helps in differentiation of brain cells and how the brain develops. So in a typical developmental condition, the neurofibromin gene actually breaks the RAS pathway so that there's like an interruption so that it's not overproducing it. However, in the NF1 condition, the neurofibromin protein is missing, or it's very low in levels. So this causes an increase in the RAS pathway. So basically it's hyperactive, and this causes the brain to shift from the normal development. So NF1 symptoms and signs for NF1. There are a lot of physical symptoms like the Lish nodules, the melanin pigment spots, and the curvature of spines, and of course neurofibromas. However, my research is more focused on the neurological symptoms. Learning disabilities, sleep disruption, autism spectrum, and ADHD are some of the neurological symptoms that are that isn't visible, but they have a really deep impact on daily life. So to understand what happens in the neurological symptoms, we're going to go into the brain. So in the brain, we have two different types of cells. One are the excitatory cells, which are kind of like the accelerator in your car. They keep sending information, they keep pushing information from one cell to the other on a constant basis, and this is what generates activity and thought. Whereas the other kind of cells are the inhibitory cells, or interneurons, which are kind of like the brakes. So these create pauses and structure in the way our brain thinks, and in the way we form memories and have our emotional state. So optimal development or optimal learning conditions only happen when the accelerators and the brakes work hand in hand. So there needs to be a balance between these two cell types so that we know that everything's working normal. Another aspect that the brain does is it has a master clock. So when I say clock, it's more like, so when, say for example, we have lunch every day at 12:30, and after a week, we are put in a room where there's no means of you to know the time. But approximately around 12:30-ish, you start feeling hungry. Same happens for your sleep as well. This is because the body has its own clock, and this is actually regulated in the brain, and these are regulated by a set of genes called the clock genes. So the clock genes give rise to the circadian rhythms that keep you in check. So when you sleep, when you eat, when you have a normal routine, your clock genes are functioning normally. And what happens in the developing brain particularly is that the clock genes also play a role in when the inhibitory neurons or the braking cells actually develop and how they develop. So it's more than just governing how your day or how your routine is, but rather builds things on a cellular level. So now you might think, why am I going into all of these? So to put it all into perspective, it's kind of like with NF1, it forms a triad. So in NF1, the clock genes or the circadian rhythm is disrupted, which gives rise to the sleep disruption that is seen. And while the clinical presentation is sleep disruption, there's internally a lot of things that goes wrong in your brain as well. So like, for example, the interneuron number, the inhibitory cell number, and the maturation of these inhibitory cells are also affected. And because of this, there's an imbalance in your excitatory and the inhibitory cells. So all of this form like a vicious cycle, which again gives rise to the learning disabilities that's commonly seen. So overall, what my project would focus on is look at how the hyperactive RAS-MAP kinase pathway and the clock genes and the braking neurons or the inhibitory neurons are actually working together that give rise to the neurological symptoms. So by decoding how sleep and how normal cellular development is during development, we are trying to move past just looking at the symptoms and rather understand how these symptoms emerge and how to manage or even target these symptoms in a better way to understand the cognitive challenges that is seen in NF1. And this is my lab. So we're working in an animal model, and we're spread across 2 different labs. And we also have Dr. Shruti Garg, who's involved with nerve tumors as well. Thank you.